In today's lecture, we are going to cover one of the most important and biggest organelles in the cell, which is the endoplasmic reticulum. We will describe the structure and function of the endoplasmic reticulum in eukaryotic cell. We will also elaborate the phospholipid synthesis. We will explain the protein targeting to the ER, as well as the integral membrane proteins. Also, we will discuss the post-translation modifications of protein in the ER lumen, and we will just talk about the protein folding and sorting within the ER. First of all, what is the endoplasmic reticulum? It's considered as a network, as you can see here, of enclosed membranes, tubules and sacs stacked on each other that extends from the nuclear membrane, as you can see here, throughout the cytoplasm. For your info, or just to remember, that the cytoplasm is everything in the cell, while the cytosol is only the fluid within the cell. So while we are talking about the cytoplasm and cytosol, we have to just differentiate between both of them. So it's considered as the largest organelles of most cells. It's a network within the cell, and it's half of the cell membrane. So as you can see, it's considered as a membrane, so it's considered also half of the cell membranes of the cells. And it's a continuous membrane, as we said, thinner than the plasma membrane. So the thickness of this endoplasmic reticulum will be always thinner than the plasma membrane. It's considered as the packaging system also of the cell, and it occupies 10% of the total cell volume. So as you can see, it occupies a large space here within the cell itself. So these are some of the features of the ER. What is the function of the endoplasmic reticulum? And in short, it's considered as the site of protein synthesis, transport, folding of the protein, lipid synthesis also, and steroid also. We will just discuss later on where does each of these functions happen. So what are the contents, or what does the ER network consist of? There are what we call cisterna, which is a flattened and unbranched sac-like structure lying in stacks parallel to one another. So these are the cisterna. They contain the glycoproteins that binds to the ribosome. So they have the glycoproteins that will attach the ribosome, so they can bind to the cisterna. There is also what we call tubules. These are the tubules. They are irregular branching elements of the ER, and as you can see, they are free of ribosomes. The black dots are the ribosomes, and these tubules doesn't consist the ribosomes. We have also what we call vesicles, which are oval in shape or rounded. They are a vacuole-like element, also free of ribosomes. There is also the lumen area, which is the area inside the membrane, which all elements are freely also communicate with each other in a fluid-like endoplasmic matrix. So all of these contents will be gathered and communicate with each other in the matrix, the endoplasmic matrix. And in this lumen, almost all of the proteins secreted to the exterior and Golgi lysosomes are initially delivered here in the lumen that we are going to talk about later on. We have two types of the ER, the endoplasmic reticulum. First one is the rough endoplasmic reticulum, which is covered by the ribosomes, as you can see, on the cytosolic surface, so it's from the outside. Its function is the protein synthesis. As long as we see ribosomes, we have to consider that the main function is the protein synthesis. It also functions in protein sorting, processing, and folding. It also functions in the assembly of multi-subunit proteins, disulfide bond formation, glycosylation, which is the initial stage that occur on specific asparagine residue through the calnexin or the calreticulin cycle that we are going to cover later on. It also functions in the degradation of misfolded protein through the proteasome pathway. So these are the important function of the rough ER, as long as it contains the ribosome also. We have the other type is the smooth endoplasmic reticulum. From its name, it's smooth, so it doesn't have any ribosome within it. What's the function? As long as it doesn't have a ribosome, so it doesn't function in the protein synthesis. So what's its functions? It's mainly lipid metabolism, especially for mitochondria, peroxisomal membrane lipid synthesis. It also functions in the detoxification of some of the drugs. The endoplasmic reticulum may pass from one cell to another through the plasma desmata in the form of desmatotubules. You can see here this phenomena, which is the plasmodesmata, happens or occur in plant cells especially. So this is a plant cell. When you focus more on their cell, you can see that the plasmodesmata or plasmodesmesis happening here between the two cells. When we focus more, we can see the endoplasmic reticulum is passing from one cell to another across this plasmodesmata. So, and they form what called desmotubules. So this is the phenomena that happens within the ER, which result in the transferring or the passing of this endoplasmic reticulum from one cell to another. So it's somehow connected between the cells. It's only not connected. It's not only connected within one cell. We can see once it's passed to another cell, it will be connected to other cell also. We will cover in the coming slides these topics, which are protein sorting, folding, lipid synthesis, and post-translation modification. First of all, we will talk about the protein sorting. In this experiment, we will know, or we will discover the pathway of the secretory proteins. When it's synthesized, where does it go? What's its pathway? This experiment was conducted in a pancreatic cells, which secretes most of their newly synthesized protein to the digestive tract. So these proteins were labeled with radioactive amino acids. These cells were labeled with the radioactive amino acids to study the intracellular pathway taken by these secreted proteins. First of all, they went in a pulse step here, which is incubation for 3 minutes with radioactive amino acids. You can see here the audio radiography revealed that the newly synthesized protein were localized on the rough endoplasmic reticulum. So here in red. So after following further incubation with non-radioactive amino acid in a chase step, the proteins were found to move from the ER to the Golgi apparatus, and then within the secretory vesicles to the plasma membrane and cell exterior. So here we can see that the pathway of the protein is first the rough ER, the Golgi apparatus, the cell surface via the secretory vesicles, and the contents will be released outside the cell to its final destination. So this is the pathway for the protein synthesized within the ER. There are two pathways or two fates for the protein in the cell. First of all, free ribosome synthesized protein and ER-bound ribosomes synthesized protein. The protein that will be synthesized on the free ribosome, it will either stay in the cytosol or will be transported to the nucleus, the mitochondria, our lovely chloroplast, which is sunbathing here, the peroxisome. While the others, and while the other type of the bound ribosome to the ER will synthesize a protein that will either retain to the ER or transported to the peroxisomal membrane, nuclear membrane, the Golgi apparatus, to the endosome, to the vesicles and lysosome, to the plasma membrane, and or cell exterior via these vesicles. Proteins destined for incorporation into plasma membrane and ER, as you can see here, the plasma membrane, the ER, the Golgi, and the lysosomes, the one with the red dots, will be inserted initially in the ER, as you can see here, instead of the lumen. So will be inserted and translocated here in the ER membrane, or inserted in the ER membrane. As you can see here, they will be inserted by alpha helical membrane spanning region, these one, and different orientation. That might be the N terminus of the protein will facing the cytosol, or the C terminus will be facing the cytosol, or it will be in a formation with a multi-pass within this ER. So this translocation, the protein will be also translocated in the ER during the synthesis. If it's synthesized by the bound ribosome, it will be co-translation, while after their translation on the free ribosome in the cytosol, it will be post-translational translocation. Both of these will happen in the yeast, while the most abundant in mammal will happen in the most abundant here, which is co-translation translocation, will happen and the mammals, and this is what we are going to cover later on. We can see that these proteins can be either co-translationally translocated after their translation, or they can be also post-translational translocated. So in the co-translation translocation, it will happen in the membrane-bound ribosome, and while this the post-translational translocation will be happen on the free ribosomes in the cytosol. So we are considering here the co-translation, which will happen within the ribosome bound on the ER membrane, which happens mostly in the mammals, while both the co and post-translational translocation will happen in yeast. So we will just focus on this one here. Let's just talk about the steps here in the co-translational translocation. So the proteins synthesis will start in the cytosol. Ribosome will translate the messenger RNA, and when protein starts to emerge from these ribosomes, it may be directed to the ER if it was destined to the ER, Golgi lysosome, plasma membrane, and for secretion, as we said before. This protein, the newly synthesized protein, has an amino acid sequence which is called a signal sequence here in red. When it is emerged from the ribosome, it is bounded by the SRP, as you see here, the green one. The SRP is the signal recognition particle, so it recognizes this signal sequence and binds to it. Then this SRP target ribosome signal complex. This is the ribosome signal complex to the ER itself and bind to the receptor. So each protein will have its receptor to bind to a specific region within the ER. So this is the SRP receptor. First of all, the signal sequence will appear or will be synthesized. The SRP will catch this signal, and then this complex, the ribosome and the signal and this protein complex will attach to the ER by the receptor. This green SRP will recognize its receptor and binds or stick to this ER. Then a GTP hydrolysis will happen, leading to the dissociation of the SRP and entering of the signal sequence to the translocon. So here you will see the hydrolysis of the GTP will happen. This SRP will be removed, as you can see, leading the signal sequence to enter the ER through this translocon. As you can see, the pink one is the translocon, which is the channel that passes this newly synthesized proteins. Then the signal sequence gets released, as you can see here. It will be released by signal peptidase, which is this here. As you can see, it will be released. It will be cut. Any peptidase mean ASE means enzyme, peptidase means protein. So it's protein that is targeting the signal, which leading to cut it and release it in the ER. So here it will be released by the signal peptidase, and the polypeptide here, the blue one, the polypeptides will be released in the ER lumen. Here you can see the animation in a clear motion if my explanation wasn't clear enough. So let's just summarize what we said. The ribosomes will will synthesize the protein. There will be a signal sequence. Once it's emerged from the ribosome, it will catch the SRP. The SRP will be cached also by the receptor to stay on the ER. After that, the SRP will be released. The signal peptide, the signal sequence will be entering the cell or the translocon. It will also be released by this signal peptidase, and then the protein or the polypeptide will enter the lumen or be released in the lumen. There are also the sorting of these integral membranes are happening in different ways also. We have the insertion of membrane protein with a cleavable signal sequence and a single stop transfer sequence. Here you can see this type of insertion or translocation will need N-terminal signal sequence, the red one. It will also need a signal peptidase, which is this one, and it will also need a stop transfer sequence, which is the alpha helix in the middle of the protein, which is this blue one, the stop transfer sequence. And it will also need a 661 transferring channel. So let's just see what's happening in this type of the insertion. As normal, the sequence is cleaved as the polypeptide chain crosses the membrane. So the amino terminus of the polypeptide chain is exposed in the ER lumen. However, translocation of the polypeptide chain across the membrane is halted by the transmembrane stop transfer sequence that closes the 661 translocation channel and exits the channel laterally. So you can see this sequence will just exit the channel laterally to anchor the protein in the ER membrane. So here you can see it's got integrated or anchored within the ER membrane. Continued translation result in a membrane spanning protein with its carboxy terminus on the cytosolic side. So this is the procedure of this protein sorting. We also have second type, which is the insertion of membrane protein with an internal non-cleavable signal sequence. So here you can see the cleavable signal sequence was cleaved by this protease. Here we don't have this thing. It's non-cleavable, the signal sequence, the red one. So it will stay, this signal sequence will act as an anchor for the protein in the membrane. So as long as you see these alpha helical shapes, so it will be anchored and integrated within the membrane. So it could be happen in two ways: the C terminus in cytosolic or the N terminus in the cytosolic. So it can, as you can see here, it will insert the cell, this non-cleavable, and it will move laterally. This end will be with no any cleavage. It will be released with the C terminus in the lumen and the N terminus in the cytosol. The other issue or the other scenario will be the N terminus will be in the lumen while the C terminus will be in the cytosol. So you can see here it might be inverted. So here the N will be outside and the C will be inside, while here the N inside and the C is outside. The last integral membrane protein sorting will be the insertion of multi-pass membrane protein with multiple internal non-cleavable signal sequences. So here an internal this this sequence will act as also anchor in the membrane. Here we will need a stop transfer sequence as the first one. The stop transfer sequence again is this alpha helix shape in the middle of the protein. So an internal signal sequence results in the insertion of the polypeptide chain with its amino acid on the amino terminus on the cytosolic side, and a stop transfer sequence then signals the closure of the translocation channel, causing the polypeptide chain to form a loop within the lumen of the ER, and translation continues in the cytosol, as you can see here. A second internal signal sequence reopens the channel. So here it reopened by this another signal sequence, and it will trigger the reinsertion of the polypeptide chain into the ER membrane and forming this loop in the cytosol. So you can see the loops within the ER and the loops within the cytosol, and it will be continuously happens, causing this multipass. So these are the three ways of the sorting of this integral membrane within the ER. Sorry, integral protein within the ER membrane. We have what we call the post-translation modifications. Once the membrane protein transferred to the ER lumen, it might be modified by the N-glycosylation. This N-glycosylation process will prevent the protein aggregation in the ER, and it will provide a signal for the sorting. So this is the importance of this glycosylation process. So as you can see here, once it enters the ER lumen, the protein will be glycosylated with the ER by the addition of 14 sugar oligosaccharides to the acceptor asparagine here, as you see the green dot, a residue. Three of the glucose residue are removed while the protein is still within the ER. So you see here, this is the oligosaccharide. It will be attached to the ASN residue here, and it will form like a dolichol-lipid carrier to polypeptide chains during their translocation across in the ER. And then it will lose these three glucose residue. Finally, this will be the modification of the protein. So this is how it's modified within the ER. We have these types of proteins. We have what we call the integral membrane proteins, that the one we just discussed. We have monotopic protein. We have single pass, we have multi-pass, and we have multi-subunit protein. So here, once you see these loops, it's a multi-pass one. Once there is no loops outside, so it's multi-subunit protein. These are integrated or integral membrane proteins. We have a peripheral membrane protein. It's on one side, and we have lipid anchored membrane proteins, which are anchored by some lipids here. They get integrated within the membrane using these lipids. Here you can see fatty acid anchor, and you can see here the GPI anchor protein. Lipids, parts of the lipid anchored proteins added in the ER, as you can see here. Example, the GPI anchor. um, lipoproteins. Here you can see the addition of the GPI anchors. This is the GPI. They assembled in the ER and added to the polypeptide anchored in the membrane by a carboxy terminal membrane spanning region. As you can see clearly here, the membrane spanning region is cleaved. Here it's cleaved, and the new carboxy terminus is joined to the NH2 group of the ethanol amine, leaving the protein attached to the membrane by the GPI anchor. So you can see here it's attached to the GPI anchor, which contains two fatty acids, chain link to the head group and the portion consisting of the mannose and glucosamine residue. So you can see here how it's get integrated within the ER membrane with these fatty acid chain instead of the polypeptide. So the polypeptide will just attach to this GPI, and it will get anchored to the membrane by the fatty acids it's already contained. Here we are going to cover the protein folding. The protein folding, the linear protein will translocated in the AR lumen will be folded and assembled into multi-subunit protein by molecular chaperones. Here these one are the molecular chaperones, which are BIP chaperone of the HSP 70 chaperone family. These are the BIP chaperones, one of the members of the HSP 70 chaperone family. And they will have also proper disulfide bond added to them by the PDI, which is protein disulfide isomerase here. So this is how it's folded. The linear one will be folded by these or will be bound to these chaperones, and the disulfide bond will be also catalyzed by the PDI, the isomerase of this disulfide bond. So as you can see, once it's get out of the ribosome, the chaperone will bind to it, and then the disulfide bond will happen, leading to the folded proteins. So this is the main procedure of the folding of this proteins. These chaperones will stabilize it and unfold into configuration until synthesis of the polypeptide is completed. The completed protein is then released from the ribosome and is able to fold into its correct three-dimensional conformation. So this is how the protein is folded within the ER. The folding of the ER or of the proteins and the conformation of the proteins goes through a quality control process. This quality controls will be held by some chaperones and sensors in the ER that will identify the misfolded proteins and divert them to the degradation pathway or to the proteasome. These chaperones are here, for example, the calnexin and the reticulum. These both chaperones types will lead to the quality control of the protein folding. So as you can see here, once it's folded, when it's released, the protein, it will go to the checkpoint. The calnexin will modify or will fold it, and it will either release it to the exterior of the cell, or it will export it to the degradation pathway within the proteasome. So it will be degraded here, and the misfolded protein will go outside the cell also. So it's either be folded and used probably by the cell that will be engulfed and sent outside the membrane, or will be degraded by the proteasome. Here another one. As you can see here, the protein emerged from the translocon. They will acquire the sugar modification, as we said. The BIP chaperones will assist the folding, as we said. Then it will go to this quality control step, the calnexin and the reticulum. They will bind to the oligosaccharide. Then, in collaboration with the protein disulfide isomerase, the PDI, as we said before, they will assist the folding and formation of the disulfide. The correctly folded protein are released, and the last glucose residue and mannose residue are removed, and the protein exists the ER and vesicles. However, the proteins that can't achieve a stable folded conformation here are sent to the endoplasmic reticulum associated degradation pathway, which is abbreviated as ERAD. The misfolded protein are delivered to the retrotranslocation channel and here exported to the cytosol. The protein are then go further modification and targeted to the proteasome for degradation. So here the correctly folded protein will be sent to the vesicles, while here it will go to the proteasome for degradation. Lipid synthesis. As we said, the lipid synthesis will appear within the smooth endoplasmic reticulum. So there are three main types of lipids within the cell: phospholipid, glycolipids, and cholesterol. They are all derived from glycerol, and they are synthesized on the cytosolic side of the ER from water-soluble cytosolic precursor, which is the glycerol. As you can see here, the phospholipid of the synthesis of phospholipid will be conducted by the glycerol. So the glycerol phospholipids are then synthesized in the ER membrane from cytosolic precursor. So it stays in the cytosolic. The two fatty acid linked to the coenzyme carriers are first joined to the glycerol, yielding to the phosphatidic acid, which will be inserted into the membrane. Phosphatase then converts this phosphatidic acid to the diacylglycerol, which is converted to the phosphatidyl choline by addition of polar phosphocholine head group. So you can see here how it's this process of the synthesis of phospholipid within the membrane. The addition of the choline, the glycerol here will be emitted, then the addition of the choline will happen, leading to the formation of this phospholipid. These are the further readings if you want to just discover more or have more clarifications. These are the animations that will just simplify the whole folding and endoplasmic reticulum topics. And this is our book, Cooper book, just to go around and read our chapter of the ER.